The helicase, which jobs to untangle DNA, moves forward to do just that on the replication fork, which is a fancy way of saying the split DNA area. There are two forms of replication. One is found on the lagging strand moving from 5 to 3, forming one half of the anti-parallel configuration. Little Lego, with the orange top, is a DNA polymerase that synthesizes all the RNA for the second half of the anti-parallel 3 to 5 strand on the other side. The telomere at the end of the DNA didn't get any RNA because it will be cut off. On the top strand, there will be something similar showing, but with more zoomed in picture. The DNA polymerase creates the Ozaki fragment, which are unconnected RNA fragments that are only combined by ligase going back and fixing it. The second kind of replication is on the leading strand, which goes from 3 to 5 and forms a solid line with no fragments in the RNA strand on a single strand binding protein. For reference, say this bottom strand is the leading strand. It would move from 3 to 5 and have an RNA primer, which is used to show where the beginning is to synthesizing it, along with a primase which would make the primers. The lagging strand is similar but has differences, such as going from 5 to 3 and its polymerase going in the opposite direction, causing Ozaki fragments. This is a close-up of a piece of a strand that has its binding proteins, making it a single-strand binding protein. The DNA splits and leaves two RNA strands, which then have connectors which wait for polymers, such as the one on screen, to make its second half. This is known as semi-conservative, since part of the original DNA is preserved, connecting to the new RNA with its origin of replication, being shown by the red Legos that attach to the main red Lego, showing the complementary base pairs of AT and GC.